Good afternoon, everyone. The third type of tissue that we are going to see today is muscle tissue. So, there are three types of muscle tissue present. So, first one is cardiac muscle, second one is smooth muscle, and third one is skeletal muscle. So, I have written something for cardiac muscle and smooth muscle that these two types are involuntary in nature, and the third type, that is skeletal muscle, it is voluntary in nature. Okay, so when I say the cardiac and smooth muscle, they are involuntary in nature, that means we cannot control these two muscles consciously. Okay, it means that we cannot control the actions of the cardiac muscle as well as the smooth muscle. Okay, but in case of skeletal muscle, we can actually control the actions of the skeletal muscle. Okay, so suppose if you want to flex your biceps, then you just think it and you can do it. Okay, but in case of cardiac muscle, which are well located in heart, you cannot increase or decrease the heart rate on your own or at your will. Okay, also smooth muscles, which are normally present in the gastrointestinal tract and some other organs also, you cannot control the actions of this smooth muscle at your will. You cannot control the peristaltic movement of the gastrointestinal tract at your will. And as you cannot control the actions of these two muscles at your will, that's why they are called as involuntary muscles. And as you can flex your biceps at your will, you can, that's why the skeletal muscles are actually called as voluntary in nature. Okay, so next, the skeletal muscle we are going to study in detail in a separate chapter. That's why we will just take an overview of skeletal muscle in this particular topic, and we will study in detail about the cardiac muscles and the smooth muscle. Even though we are going to study cardiac muscle and smooth muscle over here, you will find the mention of the cardiac muscle in the chapter, that is cardiovascular system, and in case of smooth muscle you will find its mention in almost all the chapters which you will be studying for this semester as well as for the next semester. Okay, so description or the structure of the cardiac muscle. So cardiac muscle tissue consists of branched striated fibers w- with usually only one centrally located nucleus. Okay, so sometimes you will find two nuclei in the cardiac muscle, but usually it has only one nucleus present, that too centrally. Okay, so attached end to end by transverse thickening of the plasma membrane called as intercalated disc. Okay, so these cardiac muscle cells, they are attached from one end to the another end by transverse thickenings. Transverse means like horizontal thickenings. Okay, so these thickenings, they are made up of plasma membrane and they are called as intercalated disc. So what intercalate word means? Intercalate word means to insert between. Okay, so these intercalated discs, they are the characteristic feature of the cardiac muscle, as they are not present in any other type of tissue. Okay, so it, this intercalated disc, it contains desmosomes and the gap junction. Okay, so we have already discussed about these two type of cell junctions and in cellular level of organization. So what the desmosome do in intercalated disc? Desmosome, it strengthens the tissue and it holds the fiber together during vigorous contractions. Okay, so two functions are played by the desmosome. First one is to strengthen the tissue, and second one is the hold the fiber together. So it is mentioned that during vigorous contraction. So as the cardiac muscle tissue they are present in the heart, heart is continuously beating. Okay, continuously beating means what? If you know or if you remember something from your biology classes, that heart contains of four chambers. Okay, the lower ones they are called as ventricles and the upper ones they are called as atria. Okay, so alternatively atria and ventricles they are contracting and relaxing. Okay, it means that when atria contract, ventricles are in a relaxed state, and when ventricles contract, atria are in a relaxed state. So as the continuous, and this is going on forever. Okay, till we live, this is going on forever. So this contraction and relaxation procedure, it is a continuous process. And as it is vigorous process, that means in certain cases, like if you have taken part into some kind of sports activities, so during, so it is a sport activity that is sprinting. Okay, so during sprinting your heart needs to work harder. That time pumping of the heart it happens vigorously. So during such vigorous contraction and relaxation of the heart, if desmosomes are not present, then these cardiac muscle cells they will just separate from each other, and it will lead to some problems. Okay, so heart won't function as it is supposed to be. So that's why desmosome play very important function to hold the cells together. Okay, and it also provides the required strength for the heart to beat normally. The next type of junction which is present in intercalated disc, it is a gap junction. So this gap junction, it provide route for quick conduction of electrical signals, that is muscle action potential throughout the heart. So this is important. So when we will study in further topic, that is cardiovascular system, you will come across one concept that is called as cardiac cycle. Okay, so cardiac cycle means events that happen during one beat. Okay, so when we say that there are 72 beats, heartbeats per minute, so 72 beats per minute means what? One beat is happening in less than 1 second. Okay, so if we consider 75 bits per minute, then the value of or the time required to happen one bit, it comes out at the level of zero point 0.8 seconds. So in this zero point 0.8 second, many things happen. Okay, so atrias contract, atrias relax, ventricles contract, ventricular relax. All of these things they happen under 1 second. So in order to these processes to happen at a very much faster speed, we also require certain cell junctions which will allow transport of electrical impulse from one cell to another at very faster speeds. So gap junction, as it is nothing but a gap, as it is nothing but a gap between two cells, what happens through this gap, the muscle action potential, what it is we are going to discuss in detail when we will study skeletal muscle. Okay, don't worry about this. This muscle action potential, it is type of electrical impulse. So when we say electrical impulse, it is basically electrical charge. Okay, or electricity in simple words. So this electrical impulse flows from one cell to the another. So let's assume this is cell A and this is cell B. So if muscle action potential is generated in cell A, then it needs to get transferred to the cell B. So why it needs to get transferred from this cell to this cell? Because whenever this muscle action potential is generated in any cell, the cell, it, we call the cell as the cell in excited state. Okay, so when, in case of cardiac muscle tissue or cardiac muscle cells, when it gets excited, it results into contraction. Please remember this thing very carefully. Muscle action potential, when generated, why it generates? Because of movement of sodium or calcium, okay, from ECF to ICF, that is extracellular fluid to intracellular fluid. So when this happens, movement of sodium to sodium or calcium from extracellular fluid to intracellular fluid, muscle action potential is generated in the muscle tissue. In this case, in cardiac muscle tissue, this muscle action potential, it causes the cell in which it is, or the muscle in which it is generated, to get excited, and this excitement of this particular cardiac muscle tissue leads to its contraction. So for contraction of the cardiac muscle tissue, what is required is generation of muscle action potential. And only we don't want it to be generated, we want it to spread across the entire cardiac wall. Okay, so it needs to reach to the myocardium, each and every cell which constitutes the myocardium. Okay, so how it can reach to the all the cells of myocardium? If simple a synaptic like thing is present, what is synapse that we are going to study? Synapse like things are present, like in case of neuronal transport, then it will take much greater time than 0.8 second to complete all those activities of the cardiac cycle. Okay, but as the gap junction is present, it is just a gap, or it is just a canal like structure which is present, or which is joining the two adjacent cells. The muscle action potential generated in one cell can easily get transported into the another cell. Okay, so location of this cardiac muscle is in the heart wall. So the function is, it, whatever the function played by the heart, it is the function played by the cardiac muscle. So you should understand this very clearly. Because of presence of cardiac muscle, the heart has its function, that is pumping of blood to all parts of the body. If cardiac muscles are not present, then heart will not pump the blood to all parts of the body. Okay, so this is the structure which I have just told you. This blue colored line, it is the intercalated disc. Okay, so in this intercalated disc you will find desmosomes and gap junctions. Okay, so this is actually structure, the histological section of it. So this part of it, this part you can see, it is intercalated disc. Okay, so next type of muscle tissue that we are going to consider, it is smooth muscle. So structure of the smooth muscle tissue, it consists of the fibers, usually involuntary in nature and non-striated. Non-striated means what? If we consider this is the muscle tissue, striations are like this structure. Okay, in earlier you can see, this is this thing, or this thing, this thing. It is nothing but the striations. Okay, this thing, it is nothing but the striations. Okay, so muscle, this smooth muscle tissue, it is non-striate. That means you won't see such striations when it is observed under microscope. So as it doesn't show presence of any kind of striations, that's why it is called as smooth muscle. Okay, so smooth muscle fiber is a small spindle-shaped cell, thickest in the middle, tapering at each end, and containing single centrally located nucleus. So just like that of cardiac muscle cell, it contains single nucleus which is centrally located. So gap junctions connect many individual fibers in some smooth muscle tissue. For example, in wall of intestine, you will find smooth muscle tissue which is connected together with gap junction. So this smooth muscle, it can produce powerful contractions as many muscle fibers contract in unison. Okay, so as see this in unison means what? If there are thousands of smooth muscle cells present which are connected to each other, and all of these thousand muscle cells, if they contract at the same time, then the force of contraction which is produced by the thousand muscle cells contracting at one time, it will always be greater than force of contraction produced by one or two muscle cells contracting at the same time. Okay, so you should understand this thing. So unity is important. Okay, whenever you are alone, you are weak, but when you are united, you are strong. Okay, so that's the concept is applied over here. So as a smooth muscle cells, if they contract in unison, in unity, then they will produce maximum force of contraction. So if they try to independently contract, then the contraction will be negligible. Okay, so where gap junctions are absent, such in case of iris of eye, you are going to study the structure of eye. Then smooth muscle fibers contract individually like skeletal muscle fibers. Okay, so there are basically two types we can derive from this description of the smooth muscle. That smooth muscle in which there is presence of gap junction, the type of force contraction they can produce is because of they are contracting in unison. But if the gap junctions are not present, the smooth muscle which are adjacent to each other, they are not connected to each other. So as they are not connected to each other, their contraction is also independent of each other. That means that at one point, one muscle fiber, it may contract. At the same time, the adjacent muscle fiber, it is in relaxed state. Okay, so such type of smooth muscle, it is present in, one example is quoted, that is iris of eye. So location, you can find a smooth muscle in iris of eyes. Then wall of hollow internal structures such as blood vessels, airways to lungs, stomach, intestines, gallbladder, urinary bladder, and uterus. Okay, so the function which is played by the smooth muscle is it allows motion. Okay, so constriction of blood vessel and airways, propulsion of food through gastrointestinal tract, contraction of urinary bladder and gallbladder, it all of these, they allow motion. So whatever material is present in these organs, they are propelled in either of the directions. So in case of blood vessel, when it gets constricted, the it leads to movement or motion of the blood. When airways they are constricted, it allows movement of or motion of air. When the gastrointestinal tract is constricted or contracted, it leads to propulsion of food in forward direction. Okay, so next, in case of urinary bladder, when contraction happens, it leads to micturition reflex. We call it as urination. And when gallbladder is constricted or contracted, it leads to movement of bile into the bile duct. Okay. So next, you can see over here, so relaxed smooth muscle fiber, it is as per the description, it is tapering at the ends and broader at in between. Okay, so you can see centrally located nucleus. Okay, so these yellow color thing, they are gap junctions. Okay, so this is a relaxed smooth muscle fiber. So gap junctions connect adjacent fibers, and when contracted, so it will look like this. Okay. So next, skeletal muscle. Skeletal muscle tissue, it consists of long cylindrical striated fibers. So as we have said, these cardiac muscles and the skeletal muscles, they have the striations, but the smooth muscle tissue, it doesn't have striations. So what are striations? These are alternating light and dark bands within fibers that are visible under light microscope. Okay, so what are these light and dark bands which we are talking about? We are going to discuss in detail in the next chapters to come. Okay, so skeletal muscle fibers vary greatly in length from a few centimeters in short muscle to 30 to 40 centimeters, like about 12 to 16 inches in the longest muscle. Okay, so a muscle fiber is roughly cylindrical multinucleated cell with nuclei at the periphery. Okay, so again, one difference between the other two types and this type is that it is multinucleated. In other types, in smooth muscle and the cardiac muscle, they had only one nuclei, one nucleus, not nuclei, one nucleus. But in case of skeletal muscle, it is multinucleated and the nuclei are present at the periphery. So skeletal muscle is considered voluntary. Again, one more difference between the other two types and this type, because it can be made to contract or relax by conscious control. That means at your will, you can actually control the actions of skeletal muscles. Okay, so location, usually you will find this attached to the bones by means of tendons. Okay, so the function they play, that is they allow motion, the posture, the heat production, and the protection. So four main functions are being played by the skeletal muscles. So structure, we are going to see in detail. So tendon part you can see over here. This is tendon. So this part, red colored part, it is muscle. By means of these tendons, these muscles are attached to the bones. Okay. So these are the point of attachments of the muscle to the bone. So that is tendon. So this is, you can see the entire muscle. So this is muscle bundle. So in that muscle bundle, one part is taken out. So this is the like zoom or enlarged version of this part. This looks like this when it is enlarged. So a fascicle, it is, the entire thing is called as fascicle. So this one part from the fascicle is taken out and it is then further zoomed in, so it looks something like this. So it is one part of it, it is called as muscle fiber or muscle cell. So this one part is then when enlarged it looks like this, and it is called as muscle fiber or muscle cell. So one small part from this muscle fiber is then taken out and it is further enlarged, so it is known as myofibril, and it looks something like this. So out of this myofibril, one particular part, it is zoomed or it is enlarged, which is known as sarcomere, which is composed of actin, actin and myosin filaments. So what are these actin and myosin filaments that we are going to see in detail in next chapter, that is the skeletal muscle system. Okay. So next, last type of tissue that we are going to consider, it is nervous tissue. Okay, so nervous tissue composed mainly of neurons and neuroglia. Okay, so these neuron and neuroglia you have already studied. You already know what is a neuron and what is neuroglia. So we will just take overview of both of these components. Okay, so neurons, they possess electrical excitability. That means whenever electrical impulse is generated, they get excited. Okay, so the excitement in neuron, it doesn't lead to the contraction. Please understand this very carefully. Unlike that of muscle tissue where excitement means contraction, in case of neuron, the excitements mean it leads to generation of action potential. Okay, so this action potential or nerve impulse, it is an electrical signal that propagates along the surface of the membrane of a neuron. Okay, propagates means it travels. Okay, so it will generate in the cell body. It will travel through axon and it will end into the axon terminals. Okay, so when it reaches to the axon terminal, what happens next, that we are going to see. So it begins and travels due to movement of ions. Okay, so such as sodium and potassium. So the generation of action potential, it is because of movement of the sodium ions from ECF to ICF. Okay, and action potential, when sodium moves from ECF to the ICF, it is like this. The potential changes from minus 70 millivolt to plus 30 millivolt. Okay, so when this value is reached, the sodium transport stops and the potassium transport starts. That means potassium channels open up that move the potassium from the intracellular fluid to extracellular fluid. So this plus 30 millivolt, it comes down to the again minus 70 millivolt. Okay, so what is this minus 70 and plus 30? It is the potential difference between two electrodes. Okay, so what is that we are going to study in detail whenever we will study about central nervous system. So for now, only remember this, that the action potential generation, it is because of the movement of sodium and the potassium. Okay, so this movement can happen between the interstitial fluid. Interstitial fluid, it is part of the extracellular fluid only. Okay, so interstitial fluid and the inside of neuron through specific ion channels in its plasma membrane. So for a movement of sodium, we have sodium channels. For movement of potassium, we have potassium channels. Okay, so most neurons have three parts, that is a cell body, dendrites, and axon. Types of neurons, the classification of it, we are going to study in detail when we will study the central nervous system. But now we just see the preliminary introduction to the neuron as a tissue. Okay, so these are the parts. So you know this. It contains dendrites, so it receives signal from other cells. Then it has cell body, which organizes and keeps the cell functional. Then it has the cell membrane, which protects the cell. Then it shows this particular part. It is nothing but the axon hillock. It generates impulse in the neuron. Okay, then this part is the node of Ranvier. It allows diffusion of ions. Then this cell is called as Schwann cell. So it produces the myelin sheath. Then in the cell body, you will find nucleus, which controls the function of entire neuron. Then axon, this entire part is nothing but the axon itself. It transfers the signal to the other cells and organs. So signal get generated over here. It transfers to the axon hillock, and from axon hillock, the action potential get generated, and it reaches to the this point, that is axon terminal. So myelin sheath, it increases the speed of signal because myelin sheath acts as insulator. Okay, so it insulates the electrical impulse which is passing through the axon. Okay, so the electrical impulse, how it passes through the axon, it is by it jumps from one portion to the another, from one node to the another. So it is not the electrical impulse that jumps from one node of Ranvier to the another. It is the movement of ions that happen at these particular junctions. So we will study in detail about this in central nervous system. So axon terminals, they form junction with other cell. That means they connect to the other cells. May it be the another neuron, or may it be the another effector cell, and they control the actions of the other cells. So if other cell is a neuron, then they will, whatever impulse which has reached to this particular axon terminals can cause generation of action impulse in another neuron. If the axon terminals connect to an organ, let's suppose it connects to the heart, then the whatever electrical impulse generated from the axon terminal, it will be carried in the form of neurotransmitter in the synapse. So see, in between the axon terminals and the next dendrites or the organ, there will be a gap between two. Okay, so as we know, electrical impulse doesn't jump the gap. It always flows in form, either in form of electrolytes or in form of neurotransmitter. So when electrical impulse reaches at this particular point, that is axon terminal, neurotransmitters will be released, and these released neurotransmitters will be responsible for the generation of action potential in neurons, or the production of biological activity in the organ to which the axon terminals are supplying. Okay, so next, neuroglia. Neuroglia, they make up about half the volume of the CNS. Generally, neuroglia are smaller than neurons, and they are 5 to 25 times more numerous. That means the number of neuroglia is more than the neurons. So, in contrast to neurons, neuroglia do not generate or propagate action potential. So, it has nothing to do with generation or propagation of action potential. So instead, they can multiply and divide in mature nervous system. Okay, so as we know that neurons, if they die, if a degeneration of neurons happen, it can never regenerate. Okay, that means you won't neurons don't have capacity to undergo mitosis and give rise to two new neurons. That is not possible with neurons, but it is possible with the neuroglia, as they have the capacity to undergo mitosis and give rise to the two daughter cells. And these neuroglia, they fill up the gaps which are created by the death of the neurons. Okay, so in case of injury or disease, neuroglia multiply to fill in the spaces formerly occupied by the neurons. So there are four neuroglias in the central nervous system. When I say central nervous system, I'm talking about brain and spinal cord. Spinal cord. Okay, and the peripheral nervous system, whatever neurons which are present apart from these two locations, they constitute peripheral nervous system. We are going to see about peripheral nervous system in few next chapters. So till now we will just introduce about you this concept. Okay, so in central nervous system, four neuroglia are present. First one is astrocyte, second is ependymal cell, third one is oligodendrocyte, and fourth one is microglia. So what is function of astrocyte? Astro means star-like structure. So you can see this astrocyte, it is star-like structure. That's why it is known as astrocyte. Cyte means cell. So it maintains blood-brain barrier. So very important function played by astrocyte. Provides structural support. It regulates ion, nutrient, and dissolved gas concentrations. It absorbs and recycles neurotransmitters, form scar tissue after injury. Okay, ependymal cells, the line ventricles of brain and central canal of spinal cord. They assist in producing and circulating and monitoring cerebrospinal fluid, important function played by the ependymal cells. Then oligodendrocytes, they myelinate CNS axons, provide structural framework. Okay, microglia, they remove cell debris, waste, and pathogens by phagocytosis. So they are the phagocytes of the central nervous system. Okay, then peripheral nervous system contains two types of neuroglia. That is, first one is satellite cell and second one is Schwann cell. So satellite cell, it surrounds neuron cell bodies in ganglia. It regulates oxygen, carbon dioxide, nutrient, and neurotransmitter levels around neurons in ganglia. Okay, so Schwann cells, they surround all axons in PNS. They are responsible for myelination of peripheral axons. They participate in a repair process after injury. Okay, so that's all for today's lecture. So we'll stop recording over here. So thank you. And I will upload this lecture on YouTube as well as in Clever Ground. Thank you.